Okay, today we're going to talk about about DNA replication. DNA replication is semi-conservative, meaning that each daughter DNA molecule has one old strand from the parent, so we're going to represent that with blue, and one new strand of DNA, so that's the one that's recreated. So the blue one is the parent DNA, and the purple is the blue. The purple is the new DNA. So, and these strands are also complementary and anti-parallel, meaning that the base pairs in each strand match with the adenine and thymine. The match with the adenine and thymine, and the guanine and cytosine match too. So it goes from 5' to 3', and the other one is anti-parallel, so it goes in the opposite direction. So 5' matches with 3', and 3' matches with 5'. And I just represented them as straight sticks to make it easier to see. And so DNA replication starts with the specific parts of the DNA strand called the origins of replication. So these origins are mostly adenine and thymine base pairs bonded together by hydrogen bonds. So adenine and thymine would bond together with hydrogen bonds because they have less hydrogen bonds than guanine and cytosine. So they're easier to break. So special proteins bind at the origins of replication to break apart the DNA molecule to form two V-shaped structures. So you have a circle, and that's the old DNA. But then you have this little V-shaped structure, and this is going to bend a little here, and then there's going to be a little bubble that forms, and I'm going to represent that in purple. And this is called the little replication bubble. So this purple is the new DNA. And then the black is the old. So when new DNA is formed, it comes in it expands. So this would get bigger and form another circle inside. And so they will go in opposite directions as DNA continues to replicate. So the enzyme helicase unwinds the DNA at these points by breaking the hydrogen bonds between the adenine and thymine, or the guanine and cytosine, because it is DNA, and as they go in these opposite directions. And so to prevent the newly unwound DNA, so like as these are like breaking apart here, single strand binding proteins, they bind together to pair. So they would act like in this area to like prevent these from coming back together. So like they're loose, and so they would come and attach to it to prevent these from sticking back together. So another enzyme called topoisomerase. It's called topoisomerase.